Hello students, welcome to the class of your botany. In the previous lectures, we have already done with your introduction and how the transcription happens. That's the reason I have written here transcription-02. So if you want to know it better, you need to go back, subscribe our channel so that you can actually see what we have discussed in those previous lectures so that it becomes easy for you to understand transcription. Just a general information again, transcription means your double-stranded DNA gets converted to your single-stranded mRNA and then it further goes for the translation process out of the nucleus. In the previous lecture, I said that yes, at the end of transcription, I get an mRNA with the direction of this, of course, this orientation, but eukaryotic mRNA. I'm talking about eukaryotic mRNA, of course, will consist of certain regions this is the product immediate after transcription. The moment transcription happened, I got mRNA, and because of the rho factor, the transcription stopped. Remember? So now, what happens? These are the certain regions which are present in your premature, that means it is not completely matured, premature mRNA. So these regions are, these one, let me say this, are exons, and these one are your introns. So when I say exons, these are my functional mRNAs, whereas these ones are my non-functional. That means they are of no use to us. They are of no use to us. So what happens? We need to get rid of these introns because once they are means once the mRNA which I get finally should not have these intronic regions, then only it will be able to move outside the nucleus in the cytoplasm to synthesize proteins by the process known as translation. So what happens? In this, we will do a process which is known as splicing, and this splicing will be accompanied with the help of some enzymes. So what do I get? This is what I get, because and this after splicing I will get like this. So what has happened? I have got rid of the intronic regions, so now I have got introns. So the introns will be joined with the help of again some enzymes, and at the final product that I will get a final product like this, which is my completely processed mRNA. But still this processed mRNA is incomplete. During the splicing procedure, it might tend to lose some important information. That is the reason we have to do one more step in order to make it a complete mRNA. A complete mRNA. Right now, it is not complete. So what I am going to do? I will do a process known as capping at the 5' end and tailing at the 3' end. And when I talk about your capping means addition of I'm telling you what does capping means. What does capping means? Capping means addition of methyl guanosine triphosphate. That means at the 5' end, in order to make a completely processed mRNA, which would be ready in order to do a translation process, at the 5' end I will add methyl guanosine. I'm just going to write in a bigger font so that you can see. methyl guanosine triphosphate with the process known as capping. And at the tailing means I'm gonna add polyatal. This is my polyatal. Polyatal means adenine, 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 adenine. So now what I get at the end of my complete transcription process, after splicing, what I get is my mRNA, which is completely processed. So that means if I will say on an average, I'll just give you a small flow chart so that it becomes easy for you. So what happened is that we went through a transcription process. So when I was in the transcription process, then what happened? Transcription gave me mRNA, which had exons, introns. So what I did splicing, and with the process of splicing, what I get? So first I did splicing, second I went for capping and tailing. I went for capping and tailing, and finally what I get is a complete mRNA, which is a completely fully processed mRNA, and at the 5' end and at the 3' end capping and tailing has been done. So at the 5' end what I get is methyl guanosine triphosphate, and here I get polyatal. So that means eukaryotic cells and prokaryotic cells, how do they differ? Prokaryotic cell will of course not have introns. They will not have introns. So the split gene concept is not there. Understand this? Introns are not present in prokaryotes, but yes, exceptions are always there. Archaebacteria, which is the primitive form of bacteria, it is present there. So that means introns are present in your archaebacteria, exceptions are always there, but yes, in the majority of the prokaryotes, introns are not present. Whereas in case of eukaryotic cell, of course, introns and exons both are present. That is the reason it was Richards and Phillips. Richards and Phillips, these two people gave the concept of split genes in eukaryotes. These two people gave the split gene concept in eukaryotes. Split gene means, of course, intronic regions and exonic regions, not a continuous region just like a prokaryote. So Richards and Phillips gave the term or gave the concept of split genes in eukaryotes, and they were awarded with Nobel Prize in 1993. Now what will happen? Now this completely processed mRNA, my completely processed mRNA, it is ready to go to cytoplasm for translation. So this ends up with the topic of your transcription. In the first we did what are the things or the requirements in order to process the transcription process, and in the second transcription slide what we have done, that after getting mRNA, how the processing of mRNA is done. Students, hopefully this video has been helpful for you, and if you have any doubts and any queries, what you can do, you can go back to the comment section and then you can drop your question or any sort of information that you need. Thank you so much for watching the video.